Thanks very much, everyone. So I couldn't ask for a better introduction than the previous talk. So this is great. So I guess I'll get right to it. So as most people know, okay, let me see. Is this how I navigate? Wait a minute. It's not responsive here, so I'm wondering. Oh no. It doesn't, like what's shown there is not the same as what's shown here, so I wonder if. Oh, here we go. Okay, great. Let's get to it. Okay, so as we all know, the red blood cell transfusion is one of the most common therapies. There's over 110 million donations that are collected every year. Okay, that goes into 85 million donations. So any kind of small change can potentially have a huge impact on a lot of people. And so one of the the big questions in the field for a long time has been are there differences between donors and units? Okay, are they all the same? Because right now we basically consider them even though each unit comes from a different person, once we do ABO typing, they're basically considered the same. Okay, and also what are some ways that we can measure quality? Because if are there more sensitive recipients that should be allocated higher quality bags versus for others that don't matter. And this has been looked at in many different ways. One of the most famous recent case is the INFORM trial that looked at what is the effect of storage time and what is the effect of storage time, and does it have an effect on outcome? And sort of the general answer is, well, I mean, on the whole, there is essentially no effect that transfusing younger, the blood has been stored for a younger period of time does not have any additional benefit over ones that have been stored longer. Okay, other studies have looked at what are the effects of donor age and sex, and the sort of general conclusion from that is like, okay, well, there's some minor differences. It's quite small, and maybe it may not be that significant. So that but one of the things that I think as we sort of delve deeper in this area is to start thinking about, okay, well, what is that blood transfusion really for? Okay, and if you think, okay, well, there's sort of two general classes of reasons of why you receive a blood transfusion. So one is there's like an acute situation. Okay, there's trauma. There's acute blood loss. Okay, your body is able to produce red blood cells. It's just that you lost a lot of blood, so you need that short-term infusion. Okay, and so in that case, really the benefit that you're looking for from the donation, it's only a matter of days. Okay, you get that benefit, and then hopefully your body then catches up and is able to produce red cells you need. Okay. On the other case is sort of the chronic transfusion recipients. So these are people that have chronic diseases, and they need to be on a regular transfusion schedule. And here it's a little bit different because what you really want is you want the blood to last as long as possible so that you can delay the times until the next transfusion. So reduce problems like iron overload and trachea and all sorts of complications you get from transfusion. Okay. So I think that sort of looking at it in that lens, then perhaps it boils down to the question, can we predict what the circulation time is going to be? Because if you have a unit of red cells and we say, okay, this one is going to be, it's a long circulating unit, it's going to circulate for a long time, okay, well, that one could probably go to the chronic transfusion recipients. Because those, because that's where we care about circulation time. And the other ones where, it's probably like a standard expected length of circulation time, well, those ones can go to the acute recipients because, they only need it for a few days, okay? So to them, it doesn't matter. If we can make this kind of differentiation, just longer circulating bags and regular circulating bags, then we can have much better outcomes for the chronic transfusion recipients, and maybe even increase the supply a little bit because those people are going to require fewer transfusions. Okay. And so, okay, so now you say, well, this begs the question, well, how much variability is there in the people, in the chronic transfusion recipients? Like, is there a lot of change, a lot of difference between different donors? And so we actually recently got some data to look at this from some chronic transfusion recipients. So this is a data collected by Haley Merkelly at St. Paul's Hospital. And so she had transfusion records of chronic recipients over time, and we just built like a very simple model to look at this. So essentially, what we do in this model is we say, okay, well, each bag generally is expected to give you a hemoglobin increment of 10, okay, 10 grams per liter of hemoglobin. And typically, most recipients receive 2 bags or 3 bags, and they're typically receiving it every 2 weeks, every 3 weeks, every 4 weeks, sort of on that schedule. But then, before their next transfusion, they usually take a hemoglobin measurement. So we can look at, okay, we know how much went into the patient, within reason, within some variability, and then we can see how much it's decreased. And so what we can see from here is that for some units, okay, that slope is very steep. Okay, you get you get this increment, and then boom, at the next transfusion you get a decrement. Okay, where others it's quite shallow. Okay, it seems like you get this fortunately I can't seem to run my mouse over here, but anyway, you can see on the blue one, like the second to last slope there, that's quite a shallow one. Okay, so there, the patient could probably have waited another week or even 2 weeks. Okay, so for some reason that unit has provided, much better, is lasting longer in the patient. Okay, and so if we do this, we analyze all this data, we do it for many patients, we start to get a picture over here, and this is. Whenever you run a model, you want the sort of the basic result to come out the way you think it does. And so in this case, we can see, okay, average survival time is about 10 to 15 days, okay, for each unit. And these are patients generally receiving transfusions every 3 to 4 weeks and typically 2 to 3 units, okay. But what's interesting here is, okay, so the average is where we think it ought to be. The clinician has very carefully adjusted the schedule so that, they're getting the benefit we need, that they need. But if you look, there are all these data points up high, okay? There's all of these data points over here. And some of those I think are very interesting because some of those could potentially extend the lifetime, of those, the transfusion lifetime by 50%, even 100%. So instead of being 10 to 15 days, or potentially 20 to 40 days, right? So that would be a huge benefit to those people who are, not wanting as many transfusions. Okay. So then the question is, okay, how do we identify these donors? Okay, maybe metabolomics is a way. There's I think lots of different ways to go. In our lab, we've had this long-standing research program in looking at cell deformability. Okay, the deformability of red blood cells. So red blood cells are kind of a really interesting animal, right? So they're anucleated cells. They don't have a nucleus, and they have to traverse the entire circulatory system, every minute. Okay, so and that translates to over 120 days. They go through your circulatory system about 2 million times. Okay. And when they go through your circulatory system, they have to be pushed through little holes that are much smaller than they are. Okay, so they really have to deform. And so the deformability is a very important part of the function of the red cells. In fact, what happens is as the red cells get old, they start to lose their deformability. They eventually get caught in the spleen, and then the macrophages comes and eats them up. Okay. And so there's lots of studies that have shown that if you reduce the deformability of red blood cells, they don't circulate as long. Okay, and so some of this work was done by Mark Scott, about 10 years ago. Okay, and one of the interesting things that we found, one of the interesting things about deformability is that it seems to be this accumulated effect. That deformability, cells don't get softer. It's just, their cells are made, they're pretty soft. Over time, they get more rigid. So through oxidative stress or whatever other chemical mechanisms there are, it seems that there isn't any compound that makes red cells softer. I mean, we tested a bunch of them. I mean, maybe who knows? Maybe there are. But mostly anything you do to the red cell, it's going to get more rigid over time. And so that seems to be an interesting biomarker, okay, a physical biomarker for assessing, sort of that true age or the true utility of the red cell. Okay, And so this is not a new field, measuring red blood cell deformability. People try to do this for many, many years, starting from the 60s. We put them, in a fluid. We measure it. This is an exocytometer. We try to filter them using a membrane. We put particles on them, and we pull them using an optical tweezer. We use pipettes. There's been a lot of work in microfluidics, since we've done some of it with making small structures and pushing red cells through them. And those are really fun academic studies. Okay, the problem is reproducibility. It's a boring thing, but it's really important that you take one measurement one day, okay, and you take another measurement, you get exactly the same answer. And that's the hard thing about this, is that most of these techniques do not give you reproducibility. Second thing is these measurements are hard, so you don't get a lot of throughput. You don't get to measure many cells. Okay, and one of the reasons for this is that red cells are really soft things, right? So if you just try to do simple filtering of red cells, it doesn't work that well. And so in this case, this is a quick illustration. If you put cells through a filter, they will fill up the filter, and because they fill up the filter, they change the properties of the filter, and and so that then results in subsequent cells receiving a different force. Okay, and so one way that we try to keep this is say, okay, well, is there a way that we can always keep the filter fresh? Because in the very beginning, okay, the filter, the cells are getting a very precisely controlled force. It's just over time, cells start to accumulate. That's when the problem occurs. So the mechanism we developed to overcome this is this idea of a ratchet. Okay, so the ratchet is something that has an asymmetry where you go one direction, you have like a certain response, and then in the opposite direction you get a different response. So in this case, think of a little pore, okay, but it's tapered. So instead of just a regular pore, it's a tapered pore, and the force is required to push the cell through the pore in one direction is less than the force required to push the cell in the other direction. Okay, so you get this back and forth. So if you then apply some kind of precise force, then some fraction of cells are only going to go one way, okay, through this filter. So this is kind of a one-way structure through this ratchet. And to make this thing feasible, then we make many of these ratchets. We put the cells in, and then we apply oscillatory up and downward flow at the same time with a left to right horizontal flow, and we essentially generate this staircase pattern where some of the cells are going to sort of follow this. It's going to reach a certain spot, while other cells are going to go to a different spot. So let me just show. So here's, it's quite complicated here, but it just shows you kind of what we're looking at here. So this is the matrix of pores, and the cells gets introduced on the bottom left here, and you can see that in that blow up there. We've changed the optical filters here so that the cells kind of become really dark here, and so the cells sort of progress through that staircase pattern. Let me just run this video here. Let me see if, I don't know if I can't get, okay. It's okay. I've shown this video quite a few times before. But basically, the cells kind of test out all these different structures, and some of them are going to go through, some of them don't go through. Eventually, they achieve some kind of distribution. Okay. And so the question, okay, so now they achieve this distribution, how do you analyze the data? Because typically, the cells gets distributed around, sort of 3 to 4, areas here, and typically each run, will be thousands of cells or tens of thousands of cells. Okay, but then how do you analyze the data? Well, the easy way is that you take that distribution, you make a cumulative distribution. Okay, so now it's and then if you then say, okay, if you put a crossover point, so in this case 50% crossover, then we can get a number, like a single number that describes the distribution. Okay, so basically over time, as the whole population of cells gets more rigid, okay, that population is going to move from left to right. Okay, it's going to become more rigid over time. Okay, so then we use this. Okay, so critically, like I said, a very important thing of this is reproducibility. Okay, when you take two measurements, you have to get the same answer. Okay, and so my graduate student Mel has worked, very carefully over the years to really make this possible. And so here, you have device one. So all the measurements I'm going to show you are typically done in duplicates. Okay, so there's the two devices, and we plot the result from the first one and the result from the second one, and it's very narrow. Okay, very narrow. The other thing is we can use multiple devices and measure the same sample, and generally, you get the same answer. Okay, so so then we use this to measure different donors. Of course, we start with just donors in the lab, and we see, interestingly, that there are people with different levels of different deformabilities. Okay, and anecdotally, it seems like the athletic people seem to have more rigid cells, which would kind of make sense. And then we say, okay, well, how reproducible is this? So we then ask the same donors to come back, okay, every 3 times 6 weeks apart, okay. And I know it's not a lot of data points here, but generally it seems like whatever your deformability is, seems to be a part of you. It's an inherent, seems to be an inherent characteristic. I don't know if it's genetic or environmental, but at least it's relatively stable for stable people. Okay, so then we use this to look at blood bags. Okay, and what's interesting, so blood bags are actually incredibly good at preserving red cells and preserving their deformability. And so if you look at blood bags, generally there's a gradual creep up of the rigidity. It does get more rigid, but from 0 to 42 days, okay, it's pretty good. And then if we continue this to past 42 days, to 56 days, okay, and so for those extra 2 weeks, you really start to see the deformability starts to go down quite a bit, or the rigidity starts to go up. So we had some, and there seem to be some differences between males and females, although it is, once again, a small number. Okay, it seems that the females, the red cells are a little bit more stable over time than males. But that's something that's interesting, in terms of thinking about, well, okay, are there donors that could provide red cells that last even longer than the 42 days? Okay, now, like I said, blood bags are really good at preserving red cells, okay? But if you actually want to see some kind of effect, you might want something that isn't as good, that sort of hopefully mimics a little bit of the oxidative environment of the recipients. So then we said, okay, well, could we do this in a more of an accelerated aging model, which is just instead of this optimized environment of the blood bag, we put it in a test tube. And interestingly, if you look at this, you see that some donors, the red cells are more stable, okay, and some donors, they degrade rapidly. So at donor 1 here, you can see that it degrades rapidly, okay, the cells get more rigid, whereas donor 5 and donor 8 are quite stable. So even after 14 days of storage, which we think is equivalent to about storing for the whole 6 weeks, okay, that they seem to be, the red cells seem to retain their deformability. Okay, and so this is the same data, just plotted in a different way. You can see that some the arrows here, some of them there's a big change over time, but others are not. And then we repeated there are some donors we can get repeated assessments to. We can see that okay, that general trend seems to be also associated, preserved across donors. Okay, and so. What is this building towards is that perhaps we can use red blood cell deformability as a way to screen donors, to identify those donors, okay, that can somehow produce cells that have longer lifetime, okay. And so this is sort of like a miniature summary here. Is that red blood cell deformability seems to be very strongly correlated with circulation time. And that not all units circulate for the same amount of time. There seems to be a lot of variability, even just from the simple data. And that once you have like this stable and consistent and reproducible way of measuring red blood cells, you can start to discover differences between donors. Okay. We also did a couple other. So this is sort of the study we're pushing forward, going forward. With looking at recipient material, with donor materials and looking at specific recipients. Now, a couple of things that we're doing that is going to help this is one is that red blood cell transfusion is such a safe procedure that nobody wants to deviate from that procedure, right? So it would be very helpful if we can have some other way to acquire the donor materials. And one of the ways that we can do that is to use the blood tubing, the tubing from the blood bags, because when the blood bag gets made, there's all of this extra blood that is in the tubing, okay? And generally that's either, that's sometimes used for testing, but sometimes most of the time it's discarded. And we just wanted to make sure that the material that we get from the tubing is it the same as the blood bags. Remember, the blood bag is not used right away. It's typically, at least 2 weeks later from the date of collection to the date of actual use, okay? And so we went and tested the deformability of red cells that you get in the tubing versus the deformability of the red cells you get in the bag. And it's a very good correlation. Okay? Generally, it's a very good correlation. So essentially, you can get data on the tubing, okay, and that give you the same information in the bag. So we can just, at the time of transfusion, we can just save the tubing, and then we can go and test it. Now, what's interesting about this is that hemolysis is actually not preserved. So if you look at hemolysis in the segment and hemolysis in the bags, they're totally different. The other thing that we're also studying, and my student will talk about this later on, is that testing red blood cell deformability is kind of a hard thing to do. It's something that we're set up to do, but not a lot of other people are able to do this. So we asked the question: well, if we just take pictures of the red cells, can we tell from the pictures of the red cells, sort of what is their status, what is their, to essentially try to estimate their deformability? And it turns out that, with all these great machine learning tools, you can actually get this data. Because we can get the ground truth from our deformability measurements, we can also take a picture of this and get a pretty good response, okay? So the idea is hopefully, eventually the transfusion centers of blood banks can just take their own pictures and, and analyze the data. And so we don't have to use this complicated apparatus that we have to use in the lab. Okay. So that's where things are. So I'll sum up here. So basically we have this mechanism for measuring, for sorting red blood cells to measure their deformability. And it seems like the deformability profile is consistent to the donor. And also the stability of the red cells, or how that deformability changes, is also consistent. Okay, and so if we can answer this question of, could we predict, circulation time using some kind of parameter, whether it's deformability, whether it's stability, or even images of red cells, then we can create these separate pools for chronic recipients and acute recipients, and that could dramatically improve the outcomes for chronic recipients. So I'll end right here. This is most of the work you see here is done by Mel Ismonzeta. She's here, and it's a really dated picture just before the pandemic. Yeah, thanks. I'll take questions.
That was great. It is working. That was great. I may have missed it. Are you doing these tests on whole blood or on packed red blood cells?
So if you take whole blood and you sample it, you dilute it, you mostly get red cells. So some of them are fresh whole blood collections, and some of them are blood bags that we receive from NetCAD.
Oh, okay. So I don't know what you're receiving from NetCAD. Is that whole blood?
No,
that's packed red blood cells. Because the segments are going to be obviously whole blood. That's at the time of collection, no? Or are you making segments with the packed red blood cells?
No, not the way we make those units. Those are that blood is spun, and
okay.
And then so the tubing that's going into the filled bag is actually packed red cells.
Okay. All right. It's just for clarity. Okay. Ed Prysdale up there.
Thanks for a really lucid talk. Straightforward question, technical. Temperature. Temperature's got to be a really important factor here in deformability and also reproducibility in these assays. So what do you do in there?
Yeah, I mean, we just do our experiments at room temperature. Yeah, I mean, I think, yeah, you're right. It does, it definitely would affect things. I think to control the temperature, it's, there's definitely challenges there that yeah. So I guess I think there isn't a good answer in terms. There isn't a good answer. I mean, I think we sort of do the best we can. I mean, our laboratory is temperature controlled, so we sort of kind of go with that.
Thanks, Hong.
So, Hong, another question. So it was sad to hear that you can't reverse the you can't reverse the deformability to improve it in terms of softening up the red blood cells. Is that like aging? You can't reverse it?
I think so. I mean, Jose Lopez had some idea with like mushroom-derived compounds or something like that. But I mean, anything that we tested doesn't change. Either doesn't change the red blood cell deformability, or makes it worse. Like things like pentosyphiline or like resveratrol or anything like that doesn't affect it.
I'll have to get those mushrooms. Any other questions? Cool. Thanks.
Great, thanks very much, Hung. That was really good.